I will focus on the lab 5, which is on bacterial transformation. Transformation is a kind of genetic recombination. Genetic recombination is the combining of DNA from two sources. Prokaryotic DNA from different individuals can be brought together by transformation, transduction, and conjugation. Transduction is the movement of genes between bacteria by phages, and conjugation is the process where genetic material is transferred between prokaryotic cells. In this lab, we are focused on transformation, which means that a prokaryotic cell takes up and incorporates foreign DNA from the surrounding environment. The cell that successfully absorbs the foreign DNA is called transformant. The foreign DNA is referred to a plasmid in bacterial transformation. A plasmid used in recombinant DNA research must have a few basic components: origin of replication, a site where DNA polymerase and associated proteins attach to start replication. Multiple cloning sites, a region that contains multiple different restriction enzyme sites that allow the plasmid to be cut by a variety of different enzymes. Selectable marker, a gene that produces a protein that allows the transformed bacteria to survive under conditions where untransformed bacteria cannot. Scientists often use antibiotic-resistant genes as selectable markers. AMPR is an example of antibiotic-resistant gene. In the environment that contains ampicillin, only the transformants can survive since they have AMPR that encodes the enzyme which can degrade ampicillin, while wild-type cells cannot survive. The lab experiment. The materials for bacterial transformation are DH5A competent cells, PUC19 plasmids, 4 plates containing LB, X-Gal, IPTG, 2 of them all with ampicillin. The carrier or vector of the gene to be transformed into the bacterium is PUC19 plasmid. AMPR and LAC-Z can be spotted on the plasmid. LAC-Z encodes beta-galactosidase and turns on only in the presence of galactose, which is replaced by IPTG in the plate, which acts as an inducer of beta-galactosidase. For color producing, X-Gal is required in plates. Beta-galactosidase catalyzes the hydrolysis of X-Gal to form an insoluble blue pigment, thus the transformants are shown in blue colonies. The procedure of the whole experiment is like this. First, label the tubes and plates, then obtain 1 tube of chemically competent cells and put it on ice. The cells are divided into 2 portions, in one of which PUC19 plasmids are added. After that, incubate the 2 tubes on ice for 20 minutes. Then it's 45 seconds for 42 degrees Celsius hot water bath. After the heat shock, incubate for extra 2 minutes on ice. Pipette 500 ml LB medium into each tube, and incubate for 20 minutes in 37 degrees Celsius incubator set at 200 RPM. Spin the tubes down for 60 seconds in a microcentrifuge. Remove 300 ml of the supernatant. Resuspend the cell pellets, then transfer the cell solution on each plate. With glass beads, spread solution. Stack the plates and tap them together, then put into 37 incubator overnight. The outcome of successful bacterial transformation will be like this. In the plate with ampicillin, and will spread solution of the cells with plasmids, transformants survive and are shown in blue. The experiment is closely related to real world. For type 1 diabetes, patients need to take insulin as a medication. Some patients with type 2 diabetes and other types of diabetes may also need to take insulin. Insulin helps patients manage their blood sugar levels and helps prevent short-term or long-term diabetes complications. Insulin can be produced massively by using bacterial transformation. The vector used is a plasmid that is combined with human insulin gene. The transformants are genetically modified bacteria. Lastly is the thank you note. Thanks to Free who gives out amazing lecture and being so understanding with my activities. Bye, dear friends.